Hello everyone. Today we will discuss one of the important topic for CSS syllabus, that is DNA vaccine. We all know that during COVID-19 condition, we are all looking for a vaccine. So vaccine development is the one of the important crucial aspect in our research purpose. So this is an important topic for exam point of view because everybody are looking for vaccine, so you should know that the basics of the vaccine. So today I will talk about the DNA vaccine and hope you are all doing well. So let's see the what's the DNA vaccine. So we all know that what's actually vaccine is. So vaccine actually is an antigenic preparation used to stimulate active immune response to create an immunologic memory or immunogenic memory to prevent the effect of infection. That's just the vaccine. So now we'll know the what's this DNA vaccine. So DNA vaccine, actually the DNA vaccine is a technique for protecting against disease by injecting with genetically engineered plasmid containing the DNA sequence, right? So we have to design the genetically engineered plasmid which contain our desired the targeted protein. Suppose you want to develop a vaccine against a coronavirus, so you have to design the segment of the coronavirus spike protein gene, and you insert it into the plasmid and inject it into the human body so that a human body develop antibody immune response, right? So today we'll learn about how the DNA vaccine works. Now there is no in case of human there is no available for DNA vaccine, but in case of animals two successful DNA vaccines are available in the market, but still many research are going on to develop new DNA vaccine. So let's see how DNA vaccine works and how it's increase our immune response and how it's develop memory response, right? So DNA vaccine, actually now see that the DNA vaccine is a technique for protecting against disease by injection with engineered plasmid containing the DNA sequence encoding the antigens against which an immune response is sought. So cells directly produce the antigen and causing a protective immunological response, right? Now see that in before that going to DNA vaccine, we just know about the different vaccines available right now. That's like in types of different types of vaccines, like in case of live attenuated vaccine, where we use actually the live live pathogen, but in case these are attenuated forms. Attenuated means in an inactive form, it's not able to cause disease, but it's a pathogen in a live condition. So like in case of disease like measles, mumps, polio, rubella, tuberculosis, varicella, yellow fevers, all are live attenuated vaccine. So you have to remember the which are the different types of vaccine, like MMR, measles, mumps, and rubella. Polio have two types of vaccines are available: polio savin vaccine and polio salk vaccine, right? Now tuberculosis, and another, you all know that BCG vaccines for tuberculosis, and it is also now somehow some research paper are going it's collecting that those have a tuberculosis vaccine already taken, they have a lesser chance of developing coronavirus disease. So it's actually it's maybe it's affecting some parts of the, as it is affecting the some parts of the lungs, and there is a protection in that way may be possible. So varicella and yellow fever. Now coming to the inactive or kill vaccine. So these vaccines have a so live attenuated vaccines also have some some drawbacks also available, so disadvantage also available. Like in case of sometimes it may cause as a disease, as a cause disease. So instead of protecting immune response, it may cause the disease because it's a live pathogen. So live pathogen may turn out into a and cause the disease. so condition. So these are possibilities for live attenuated vaccine. Disadvantage, and but advantage is that it's, as it is a live form of the pathogen, so it's increase the immune response much more than other types of vaccine, right? Now in case of inactive or kill vaccine, inactive and kill vaccine, here actually the pathogen in a or treated in formaldehyde or different types of stress condition, and then we develop inactive or kill vaccine. So it's like in case of cholera, influenza, hepatitis A, polio, Salk vaccines are inactive or kill vaccine. But it's have an advantage is that there is no chance of developing disease after taking the vaccine. So it's a, and disadvantage is that there is a less immune response, or it will take time to develop the immune response. So that's needed booster dose for developing the immunity, right? Now coming to toxoid. Toxoid actually here actually the part of the different part of the toxin like exotoxin or endotoxin actually used as a in case of toxoid, like in case of diphtheria, tetanus, or a toxoid vaccine. Subunit vaccine or inactive exotoxin like hepatitis, pertussis are subunit type vaccine, and other type of vaccines like even DNA vaccine and vector vaccine. We'll see how DNA vaccine works. And another is you have to know that the dose, so age of dose, possible dose, like in case of BCG vaccine actually given as soon as possible after birth. Polio vaccine is given after the childbirth of 6 week, 8 week, and in 8 week possibilities. DPT, diphtheria, pertussis, and tetanus, these vaccines are only given after the birth of 6 week, minimum the age of the 6 week after birth. So a typhoid vaccine is given after 6 months, measles vaccine 2 years, mumps vaccine 12 to 18 months, rubella vaccine 9 to 12 months, and hepatitis vaccine as soon as possible after birth, right? Hepatitis B vaccine is available. So and hepatitis vaccine, A vaccine also available. So hepatitis vaccine as soon as possible after birth, right? Now we'll see how to construct for DNA vaccine, or how DNA vaccine works. These are the new this image actually taken from a recent paper, that is Nature Communication, Smith et al. 2020 paper. So you can see that here is a they are trying to develop the vaccine for coronavirus, and they are targeting the SARS-CoV-2 spike protein. And what's the engineered? We all know that during a plasmid have a minimum construct is that it must have an origin of replication site, it must have an antibiotic resistance site, it must have a multiple cutting site or cloning site, right? Now here you can see that, so this is spike protein gene actually insert is, these are the insert actually taken from spike protein, right? Spike protein. And now this spike protein actually, this plasmid actually now injected or in the animal model, and by use of the gene gun or different micro injection technique, so that it will enters into the cell and it will develop a cell, and it will use the host machinery or human promoter. It will transcribe and translate under the human and regulation, or human regulate different genes, or different genes, and it will produce the proteins, and spike protein will be produced. Now this spike protein will activate the different types of B cell and T cell, and then it will develop the memory response of or antibody response, right? Now another is that there is a SARS-CoV-2 OL also for spike protein. These are the vector construct that's actually developed for DNA vaccine. Now we'll see the how DNA vaccine actually works. Suppose these are the these are vector containing our gene of interest, like in case of spike protein gene. So spike protein gene, now we are injecting this vector or plasmid DNA containing DNA into the host cell, or in case of human body, now it will enters into the human cell, and this gene will transcribe under the promoter of human gene regulating promoter, and then it will produce it will transcribe and translate into the different types of protein. These are antigenic protein will develop. That's actually spike protein will develop. Now there is a two types of pathways are there. Actually, it's a priming cellular response, cellular immune response. These are cellular immune response, and these are humoral immune response. Cellular immune response, like in case of here, the local APC, that is local antigen presenting cells, are taken up that at plasmid, right? And it will transcribe and translate into protein. The protein will express in the surface. Now surface actually attract the cytotoxic T cell. Now cytotoxic T cell activate the activated cytotoxic T cell, and help to increase the different types of cytokine receptor will express. These are the cytokine receptor of the T helper cells in the site, and it will act. These are also T helper cells, and these are the T helper cells, and these are the TH1 cytokine, right? These are the cytotoxic T cells. These are cytotoxic T cell or TC cell. Now this also, like in case of this also cytotoxic, these are T helper cells or cytotoxic T cells. Now this will activate the antigen binding, cause the cytotoxic to the cell and multiply and attack the inoculated cells. These are actually culture cells, and now the after inoculation it will, some of the T cell become memory T cell, and that we know that cytotoxic T cell actually kill the inoculated cells, right? Now here another is the humoral immune response pathway. Here these are the local antigen presenting cell, local APC, local antigen presenting cell. Now local antigen presenting cell, these are the receptor, and and it will same way transcribe and translate, produce protein. This protein will express in and bind as a acts as an antigen. So antigen and a Th cell will be activated, and after Th cell activation, antigen binding trigger the release of cytokine. These are the cytokines that release. These cytokine are known as Th2 type of cytokine. Th2 type of cytokine, we know that there is two type of cytokines are there, Th1 cytokine and Th2 cytokine. Generally Th2 cytokine leads to immunological suppression, right? And Th1 suppressor actually and pro-inflammatory cytokine. Now here cytokine activate this Th2 cytokine. Now Th2 cytokine activate the B cell. Cytokine helps to activate the B cell. Now here the antigen, these are the spike protein antigen. Suppose these antigens are bind into the B cell surface, and B cells also activating from the Th2 cytokine. Now the B cells are different, become activated B cells. Activated B cells produce antibody. Several types of antibody will generate. All antibody will generate, and this antibody actually helps to neutralize the pathogen, right, or virus or pathogen. And now another is the memory B cell. This memory B cell also produce in that way. So in that way the actually vaccines, DNA vaccines work, right? Now first of all, so first of all is that you have to construct a vector. Vector should contain your insert. Insert is that the gene of interest. That gene of interest, suppose you want develop in case of against the spike protein, antibody against the spike protein, so you have to insert the spike protein into the vector, spike protein gene into the vector, right? And the vector construct will be new construct will be possible, and this engineered plasmid, or it's a laboratory generated plasmid, will be injected into the host cell, and then host cells will produce the protein, spike protein, and this spike protein acts as an antigen, and it will develop the immune response, and in that way the DNA vaccine works. But there is a less chance of successful of the DNA vaccine because of several reasons are there. So we'll see next in the advantage and disadvantage of DNA vaccine. Now, so first of all is that no risk of for infection. In here chance of infection is less. Suppose in case of live vaccine, and there is a live attenuated vaccine, there is a chance of reinfection because pathogen may turn up into a new pathogenic form, and there is a chance of possibilities is there. But in case of DNA vaccine, there is no risk of infection. So antigen, here is a antigen presenting cells, antigen presentation by both class 1 and class 2 MHC molecule, and that's why it will activate both humoral as well as cell mediated immune response. So there is a chance of greater immune response to develop, right? So polarized T cell response toward Th1 and Th2 type immune response focused on antigen of interest, so antigen of interest, because here antigen, and that's the spike protein is never be modified form. It's a actually whatever the pathogen actually contain, the spike protein will be, suppose that you are engineering, these are the vector construct, these are the your insert, and this insert will express, this instruct will, these are the vector part, and this actually generates some protein, and this protein actually whatever exactly the pathogen have. That means the immune response will be much higher than other type of response, because there is no modification, no changing occurs, and as exactly pathogen express its surface, it will present in the host, or it will generate into the human body, and it will generate the antibody, right? Another is the stability for storage and shipping. There is no, it's a highly stable, no refrigeration is needed, so that's why it's a low cost effective in that response of storage respect, and long term of persistence of immunogen, right? Now in case of, you can see that in vivo expression ensure protein more closely resembles normal eukaryotic structure with accompanying post-translational modification. The immune response is therefore directed to the antigen exactly as it is expressed by the pathogen. That's why I'm talking it that these are the actually pathogen will be whatever the pathogen actually have, it will express in the same form, and then that will increase more immune response, or and there is a higher chance of working the vaccine in the real time, right? And the DNA vaccines induces both humoral as well as cell mediated immune response. DNA vaccines, we have seen that there is a this DNA vaccines actually, these are the cell mediated immune response, and these are the humoral immune response, these are B cell mediated antibody response, and this pathway for cell mediated immune response. So DNA vaccines actually working in both pathway, increasing the cellular immune response as well as humoral immune response, right? Now now another is that the DNA vaccines also induce prolonged expression of antigen, enhancing in the induction of immunological memory. So DNA vaccines also, because it's there is no chance of degradation, right? And so that's why it's as it in a DNA form, in the, and it will transcribe, and then it will translate, and then it will form protein, right? So that's why this a prolonged expression of the, and as it is a in normal form, there is no chance of degradation. So DNA vaccines also induce prolonged expression of antigen, enhancing the induction of immunological memory. Now we'll see that disadvantage also there. There is a limited to protein immunogens only, because if it's a polysaccharide, suppose the antigen is maybe polysaccharides also, but it's not possible for vector construct or a DNA construct. There is no chance of a polysaccharide, because it will first of all it will the transcribe, and suppose it's a, if a gene of interest protein is here, suppose these are interest of construct is there. Now this construct actually transcribe, and it will produce only protein, so there is no chance of polysaccharide. So polysaccharide will not be immunogen, acts as the immune or antigen. So limited to protein immunogens only. These are the disadvantage. Risk of affecting genes controlling cell growth, and there is an important factor is that it may integrate it into the normal genome, and it will cause cause uncontrolled proliferation, maybe cause some genetic mutation called possible, and it may leads to cancer also. Several other disease possibilities is there, and risk of affecting the normal controlling genes. So there is also this risk of affecting genes controlling cell growth, and these are the and possibility of inducing antibody production against DNA. Now, another possible chance is that the possibility of inducing antibody against DNA. Now, and there may be chance of autoimmune condition, and may be possible. So, possibility of tolerance to antigen and produce. Another possibility is that and potential for atypical processing for bacterial and parasite proteins. Right now, we'll see the previous questions. What's actually DNA vaccine related questions are came previously, so we'll see that how the question is came. So read the question first. So first is that DNA vaccines offer several advantages over the existing vaccine approaches. Which one of the following statement related to DNA vaccine is not correct? Now see that first option, the immune response is directed to the antigen encoded by the DNA, able to induce both humoral and cell mediated immune response. Right, we have seen that. Yes, these are correct. Immune response, because DNA vaccines works in both pathway, humoral as well as cell mediated immune response. Right, now DNA vaccine can induce prolonged expression of antigen, enhancing the induction of immunological memory. Yes, these are also possible. DNA vaccine induce prolonged expression of antigen. DNA vaccine could remain stable and potent for long time without refrigeration, eliminating the challenges of storage and transportation. Yes, this is also possible. And DNA vaccine construct can be engineered to carry several antigen to affect host and replicate in neuronal cells. Neuronal cells. Acha. Now these are the actually possibilities that uh, it's a several antigen cannot be possible, because your if it is a your gene of interest, suppose only one antigen can be possible one time, several antigen cannot be possible. Another is that infect host to infect host and replicate in neuronal cell, these are not possible. So option 4 is the answer for not correct. So option 4 is the answer. Hope that will make clear. So first, in summary, what we can say that the DNA vaccine actually, it's a because we develop the antigen, antigens are induce the immune response, and then antigen develop memory response, and that will act as a vaccine in normal condition, right? But in case of DNA vaccine, what's actually done, the gene of protein, this protein particular, we target the protein and it goes into the gene, and we from the like bioinform different bioinformatics tools are there, we construct a particular gene segment and design the gene segment into the laboratory condition, and then construct into the DNA in the vector insert the gene of interest into the vector, and this vector then in the liposome form, or in the in a gel gun, we can introduce into the host cell, either in the muscular injection or in the in possible of gene DNA or gene gun can be possible. It will be introduced into the host cell, and then it will insert into the host cells, use the host cell machinery, and then it will produce the normal pathogenic protein, and this pathogenic protein will develop the immune response. It will work via two pathways, humoral as well as cell mediated immune response, and another possibility is also develop the memory B cell response. Memory B cells, it will, so it will protect protect us from future infection. Another important is that DNA vaccines, still now no DNA vaccines are available, but there is several research are going on the basis of developing, like in case of AIDS, malaria, there is a chance develop there is research is going on for developing the DNA vaccine. But in case of animals, like others animals have a some two DNA vaccines are available. And still now many research is going on for developing the coronavirus vaccine in terms of DNA vaccine. So these are important, helpful for your exam point of view or also question paper, because these are first challenges to develop the vaccine right now. So hope this make helpful. Thank you.